Hi, my name is Jakob Troidl, and today I present Vimo, a visualization tool to search and analyze connectivity motifs in large brain networks. Neuroscientists can reconstruct three-dimensional models of neurons using high-resolution electron microscopy to better understand how the brain processes information. For example, here we show a set of neurons in the central brain of the fruit fly. Those neurons have complex three-dimensional shapes and form large connectivity networks. But what do we actually mean by neuronal connectivity motifs? Brain networks can be interpreted as directed graphs, where nodes are neurons and edges are the connections between neurons, so-called synapses. Motifs are small repeating subgraphs that can be seen as building blocks of computation in the brain. For example, new research shows that specific motifs in the brain of a fruit fly mediate responses to temperature. But finding and analyzing these motifs is inherently complex. The most recent connectome datasets contain more than 50,000 neurons and over 100 million synapses. For such large graphs, motif discovery is computationally expensive. Also, scientists want to explore not only the high-level connectivity between neurons, but also dive deeper into how neurons actually form a motif in 3D space. Early motif analysis tools for connectomics required writing complex programmatic graph queries in languages like Cypher, or focused on efficient parallel algorithms to enumerate subgraphs in large networks. Other related work shows how to visualize motifs in dynamic networks or visually simplify motif instances using glyphs. However, those visualization approaches do not scale to complex spatial and static graph layouts that we're seeing for connectomics data. Also, previous tools to analyze neuronal connectivity were focused on small connectomes and therefore did not scale to large connectivity networks that Vimo supports. Now, what exactly are the goals of neuroscientists when they want to study connectivity motifs? Over the course of 1 year, we held a set of semi-structured interviews with domain scientists from the Harvard Center for Brain Science and HHMI Janelia Research Campus. We identified 3 domain goals. First, they want to search a connectome for instances of a given motif. Ideally, they can map certain biological attributes to the structure of a motif. For example, by specifying the type and brain regions of neurons and synapses, they can filter for motifs involving neurons with certain connectivity preferences and functions. Second, they need to correlate the motif connectivity with the neuron's morphology. Clearly understanding how the motif connectivity is embedded in the 3D structure of neurons is crucial to understand the function of neuronal circuits. Third, they need to understand the spatial relationships and interconnections between different motif instances. For example, in this network, 3 triangular motifs are arranged in a grid-like structure. A better understanding of such details helps to study the flow of information through a neural network. Vimo addresses these challenges by allowing users to interactively search and visualize arbitrary connectivity motifs in large brain networks. Users can sketch a motif and define biological constraints like neuron types and connection strengths. Motif constraints make queries more biologically meaningful and also reduce the search space of the motif discovery algorithm. Here, we show the motif drawn in task 1 of the case study in the paper. For each sketched motif, we also display how often it occurs and if it's over or underexpressed in the data. As a next step, users search for instances of the sketched motif. The list view shows found instances, and users can select and inspect the results in more detail. To better understand how neurons form a motif, we've designed a novel focused and context approach that gradually highlights the connectivity in a 3-dimensional dataset. Our approach consists of 3 main steps. First, users can slowly prune all neuron branches that are unrelated to the motif highlighting the core motif connectivity. Second, by dragging the slider further, Vimo untangles the neurons by transitioning to an exploded view. This allows users to inspect complicated neuron morphology with less occlusion. Third, we hierarchically cluster and bundle connections between neurons to highlight synapse clusters. Additionally, Vimo allows graying out unrelated neuron branches and highlighting brain regions to provide spatial context for neuroscientists. Neuroscientists also use Vimo to study the spatial relationships between different motif instances to get a more holistic view of information processing in the brain. On the one hand, Vimo supports a neuron-centric analysis by showing if a neuron forms the same motif with different synaptic partners. On the other hand, users can also explore synaptic pathways starting from a seed motif instance. They can trace pathways built by repeating occurrences of the sketched motif by setting specific neuron IDs as node constraints before querying the brain network again. For in-depth statistical analysis, experts can quickly open neurons in other tools, for example, Newprint Explorer. The source code, a live demo, and detailed tutorials about Vimo are available on GitHub. In our user study, we've shown how our tool lowers the threshold for neuronal motif analysis, and we hope that the neuroscience community will adopt Vimo into their workflow.